So friends, today we are going to discuss about organ of electrohomeopathy. So basically, today we are going to talk about cohobation. Okay, in organ of electrohomeopathy, we are going to talk about cohobation. So basically, there are multiple methods. So to preserve the essence or the force, the bioforce which are present in the plants in ancient times, the number of scientists, number of persons will save that essence from the plant. So basically, here not we are not discussing all the methods. We have number of methods like I will explain one old method that is we can call cold fermentation method or cold fermentation, mostly cold and hot. There are two types. Hot, we have to boil it, and we have cold fermentation method. Both methods are utilized by old ancient people and old ancient scientists. Okay, so they preserve that what are the bioforce which is present in the plant. Okay, basic idea is for every person is to preserve the bioforce of the plant. But actually, what happened that along with bioforce, we know that there are the negativities and positivities present in every part of the human being, animals, plants, every in every creature. There is positivity, there is negativity. So what we need positivity. So along with that bioforce, the negative force also will consume with that help of these processes. Okay, so which can help the human being. 90% it will give the effectful results, but there is a chance of 10% the negativity which is present in the plant may be transferred to the human being. Okay, so basically, the pain, this idea is cut down by our Count Caesar Matty. He absorbed every types of cultivation fermentation process. Okay, so like olden days, we preserve achar, pickles, and everything. So we can see that our nani, grandmother, grandfather, they preserve olden days things. Okay, now there is we are unable to see those things. Okay, because everyone is having packed food, packed vegetables, packed meat, everything they are taking from the market, buying, and they are eating. So that's very harmful to their body. Okay, so the fermentation, the coagulation process is not a new, but is old theory. But Count Caesar Mattei took that theory and he established his own ideas to remove the negativity from that process. Okay, so there's the reason. This you can see that the older method, they will take all the parts of the plant or vegetables or anything in the active period of the season. Like we have number of plants we can only available in the month of November and December. Some plants can available in winter rainy season. Some plants will available in summer season. Likewise, they will took that plants, and what they do, they will cut the pieces of the plants, and what they do, they will put into the distilled water. Okay, and they will give the temperature of 95 degrees Fahrenheit or 35 degrees Celsius. So it is the water is above, and the whatever the material we put, it is below the water. You have to see that. Okay, what happened after the boiling processes? The boiling temperature will reach to the 95 degrees Fahrenheit. So what will happen? The essence, what is present in the plants, will start releasing from their own material. Okay, so one time will come, the whatever material we put it on in the water, it is not able to release. So what will happen at that time? what we will do, we will remove that plants, and again the process will continue. So likewise, they will prepare the uh, hot fermentation, hot coagulation process. And again, we have cold fermentation process. In olden days, they are also used. So what will happen? The old cold fermentation, they will not boil the plants. They will put the plants into the water, and they will put in the not in the closed room for 40 days, 60 days, and then after that they will release that, and we will get the spygeric essence whatever need from the plant. You can also try in your home cold fermentation. Like example, I will give that you can put curry leaves, because curry leaves are very good for hypertension, okay, and stress release problems, okay, hair fall. That is very much given in research papers also we had. Okay, so you can try curry leaves, and we have other plants also. You can try cold fermentation. You can put in the very glass jar. You need glass jar, one beaker glass jar, and you have the tight uh, cork or a tight cap, okay, and you put some leaves in that, and leaves are below the water. Water is full, okay. You can take something 500 ml or 250 ml. In that you can put all the curry leaves, and you put in and crock it, and you can cover it, and you can put in a dark room, okay, where the sunlight doesn't get onto the glass jar. Okay, and you can remove after 40 days. So you can observe what the essence of the curry leaves will release in that distilled water. So after that, you can try it like that also. You will get also results, some results. So basically, it's upon us what type of cohobation or specific persons we can use. Okay, these spygeric essence are, they are colorless. Some are colorful. That some colorless, some odorless, some are tasteless. Okay, basically, what the process is, the this olden process is using all the pharmaceutical companies, but the electrohomeopathy process is not used by a number of companies. So I visited personally, and in the seminar also, to the Rabison. Okay, so they are providing only electrohomeopathy process of making spygeric essence, which are used by our doctor consider medic, not other. I believe that, okay, it's upon you. So I believe that they are 100% electrohomeopath, other than other companies which are providing electrohomeopathy medicines. They are using old fermentation method. They are making spygeric essence, but not in Matties method. Okay, they are providing you in their waves or old ancient type of cohabitation process. So there's the reason what happening, that I told you that in the beginning, the cohabitation process which are used in ancient periods, what will happen? They have positivity and negativity. So we are taking positivity along with negativity. So there's the reason they have some side effects. Uh, if you give other than remedies, okay, the patient will have in the homeopathic they will call aggravation, okay, but in electrohomeopathy we'll call that symptomatic. We can call it not aggravation but symptomatical. High the symptoms are increasing. Okay, that's can term use. The not aggravation is there in electrohomeopathy. Aggravation is seen in homeopathy or other medical systems. Okay, side effects, aggravation. Okay, so let's talk about the process which are used by Rabison and original electrohomeopathy practitioners, or Matties method. Matties method, the process making of spygeric coagulation process. So first of all, what's one important thing we have, that we have to collect good quality and good quality herbs. because good quality herbs only will give you result when you are practicing, table practicing. Okay, this is very important. So what's good quality herbs are collected. Okay, and also in good mode. Okay, one leaf is breakdown and one leaf have some insects are there, so you are taking those leaves also, so it will have negative energy. So you are making combustion spygeric essence. What happened, the negative energy is also you are taking from that plant. Okay, first is collection and cleaning the herbs, very very important. So what will happen, collection of herbs will be in particular region of the year. So I told you that the particular area, the particular region of the year have some specific plants we have to collect, and we have to collect in fair and fresh mode. Okay, and we have to take that plants from that area to the collection place within 6 hours. Okay, and so in electrohomeopathy, number of most of the 106 or 109 plants are seen in our India. Okay, all the plants are collected in fresh mode. Okay, from February to November. Okay, every year. So most peak region is 15th May to September 15th. So this is the peak where you will get all the fresh and very good plants. Okay. And moisture, whatever moisture is present in the plant will not exceed more than 20%, because moisture is very important, because that moisture will have the bio force, bio energy. So we, if we want to get 100% quality and quantitative remedy, so what we'll need, we will get, we need 80% moisturization. Okay, and we have to collect in healthy place, healthy area, healthy weather. So if there is a pollution, all the dumping yard is there, and there is a nala, very dangerous chemicals are released. So now you will get the negativity from that plant. You doesn't you get, you doesn't get positivity. Okay, you have to remember that. Okay, so now the collective plants will reach to the laboratory, and what will happen? We have to cut down the plants, we have to clean that plants. Okay, and whatever flowers, roots, we have to handle properly. Okay, and whatever vehicles we are using, we take very hygienic condition. So then only we'll get 100% material from that plant. Okay, now you have to clean the herbs. After collecting the herbs with a clean, neat, hygienic process, flowers, roots are very handle it very easily can handle. How properly we handle that herbs. Now we reach to the collecting area, and we have to what to do? We'll clean the herbs. You know, and we'll clean the fresh and clean water. Okay, and we'll clean with drinking quality water. Okay, okay. There is no contamination. We have to see that. Okay, and whenever we are using water, we doesn't use we you have to use insect free processing. Okay, we have to free the plants from dust, all types of things we have to uh, take care, and we doesn't use chemicals and any things to clean the plants. So this is very important. We have to clean the plants with only quality water. First is, now the plants are ready, the plants are clean. Now what we will do, we will take the sulfur from the plant, means the main contents of the plant. Means we have the body, means we have the body, flesh, we have the bones, we have the soul. These three combination is necessary for every human being. Physical part, soul part, and we have nutritive part. Here, extraction of sulfur. Extraction of sulfur. This is a process of extraction in which the fresh plant to be used. The whole part of the plant is cut into small pieces. So what will it? The fresh plant whole to be used. The all the plant to be used. They will cut into the pieces and put into a container. In the container, the steam will pass. Steam will pass. So what will have? The volatile contents, the main contents of the plant is taken from that help of steam. Okay. So this is the sulfur. This sulfur means the pran vayu, means the main content of the plant. It is not a chemical. Okay. So first is extraction of sulfur. So we will see in a video also. Okay. I will see you in the video. I will put my video where we went to the Rabison company. Next, extraction of mercury. This is very important, and this will take more time. Okay. After extraction of first part, that is sulfur, the rest part of plant from its part to be taken to the second step. It is put for the fermentation at a temperature of 22 degrees Celsius to 35 degrees Celsius. It varies plant to plant. Next, we will put in a container, and whatever the part of the after extraction of sulfury, the whatever remaining part is there, what we will do, we'll remove, we will put in a temperature 22 degrees to 35 degrees. So here fermentation will happen. So at least it will take 17 to 31 days. It will also depend upon the plant to plant. So fermentation, the liquid part to be filtered out and rest solid part to receive to be allowed for shade drying. What will happen? Liquid part will take out, and whatever solid part is there, we will shade dry. We will dry into the shading area, not in the sun. We will dry in the shade area. So now the liquid filtered to be distillation for 7 to 10 times to get the second part, that is called mercury. Actually, this is the alcoholic part of the plant, also known as liquidated of the plant. Here the mercury term used for the liquidate part of the plant or alcoholic part of the plant. This term is not used for any type of chemical. So here second part we will get the after distillation number of time we distill, then you will get second part, that is mercury. First part is sulfur, second part is mercury. Okay, this is liquid part of the plant. First is solid, no, not first is vayu sol, now is liquid, and third is the hard material which we dried in the shade we will get now. Okay, next is salt. Next is salt. The solid residue after shade drying to be burned and ash to be collected after incineration. The ash to be calcined to a special apparatus with 40 to 600 degrees centigrade. So what will take? It will take 7 to 10 days to extract the salt part of the plant because it contains carbon, okay, which is very dangerous for the body. So if you use the salt part, so what will happen? It will if you have to we have to ash it. So we need 40 to 600 degrees Celsius. So after calcination, the calcinate material to be dissolved in distilled water. So it is dissolved in distilled water and put dissolvent on a constant heat 25 degrees Celsius to 35 degrees, 350 degrees Celsius. Then the whole distilled water is vaporized and the powder material to be collected from the pot. This is salt of the same plant. So again, distillation after distilled water is mixed. Again, 250 degrees Celsius to 350 degrees Celsius Fahrenheit is given. Then the distilled water is vaporized. So what is remain? Salt. The powder is remain. We'll call salt part of the plant. We have sulfur, we have mercury, we have salt. Three things are there. Now we will mix, and we will mix all these three things and put into gestation period, that is gestation 10 to 16 days, and distillation will happen 7 to 10 times. So the end part, what we will get is spygeric essence, crude form of the material, or we will call D1. What we will call it is called D1. Very important. Okay. So here sulfur has the color like water, taste like water, or sometimes little bit so. Mercury, color, fermenting matter are brownish to deep rusty brown, but after distillation it become water. Taste, bitter, some plants sour. After distillation, become little bit bitter and sour. Okay, order is decay and matter distillation. Order becomes like fresh plant. Salt color, white and off white, some tight time light pale, taste sour or sometimes bitter. So this is the physical properties. Okay, after that, so step one, extraction of sulfur. Step two, extraction of mercury. Step three, salt bar. Step four, process of gas station. Then we'll get D1. After D1, they will prepare D2. After prepare D2, D3. D3, they will use us. Okay. So देख सकते हैं आप, you can see that this video. So this is the, okay, this is the second part. You can see that this is the, after taking sulfur. First we will see first part. Yes, this is the first part. This is the whole plant. Okay, from that here steam is going, and steam is coming here from this part, and you can see this is the whole plant. Okay, after that the volatile parts of the plant can be reaching here and it is stored here. Okay, it is storation of the volatile part. First part is sulfur. So this is the first part, sulfur. Okay, and after fermentation, the the liquid part, the liquid part of the plant will become this color. And what will happen after boiling? Okay, how much centigrades? They will 20 to 35 degrees Celsius. It will take 17 to 31 days. Okay, 7 to 10 times. Okay, great. Okay. So this will take 7 to 10 times. After that, this is, you can see that this is collecting part of the second part, that is mercury. So again, please see the video. This is the lab of Dr. Sanjeev Sharma sir, which who worked very hard to bring main electrohomeopathy medicines to India. He is doing his job very well. So basically, this is coagulation process. Actually, so you can see this third part. Okay. You can see this third part is prepared here in this area. You can see. You can see that this is the area. This area, this container. This will make ash, and then after this, then after they will do distillation process. Then after again, it will become uh, salt. Okay, this is the machine. Okay. I think complete mission is not able to. Here also one mission is there. Here also one mission. This is the lab. Okay. So this is the process of making coagulation. I hope you understood. So any doubts? Please go to doubts.